the point that concentration of transcription factors matter, even the concentration of the same transcription factor. So what we're going to look at is a particular protein called Kruppel. Now it's discovered by German, obviously. Okay, now it was discovered in fruit fly embryos. Of course, fruit fly, especially when you look at a fruit fly larva, in other words a maggot, you can clearly see that wormy segmentation pattern. Ever since we came up with annelids in this planet, everything higher than that has a segmentation pattern during development. We have that ourselves as well. It's very clearly seen when you look at things like annelids and, segmented worms. You can see, if you look at the body of an insect or a crustacean or something, you can see that segmentation pattern. If you look at our development in early embryonic life, you can see that segmentation pattern. In fact, did anybody see a couple days ago something that looked like it was a series of segmented something or another? Yeah, what did they call those things? Those blocks of dense mesoderm. Solites, yes. You see a segmented pattern. And if you look at like development of our central nervous system, things like that, you see that we have somites in our development as well, and what have you. So we even though we don't look so much like segmented worms, unless you happen to be in Congress. Parasitic segmented worms, by the way. Leeches. Okay, but anyway, we see our segmentation pattern developing in early embryonic development. Well, so you look at fruit fly embryos. The Kruppel gene is a transcription factor that activates a whole bunch of other transcription factors, and it and many other these kinds of genes ultimately activate transcription of different Hox genes. Recall, we said the Hox genes are the ones that are expressed in various segments and organize the embryo from anterior to posterior. That's something you find everywhere from sponges all the way up to us. So, they're in a class of what are known as Pairrule genes, and they're called that because these are expressed in bands of two future segments in the embryo. So we see like Kruppel might look like this, you have some here, you have maybe another band here, and things like that. Okay, now, so that's what it is. So it helps to organize the anterior to posterior part of the embryo. Now, what happens is, here's an example of a protein that binds to an enhancer that's quite close to the promoter. And this protein directly interacts with the basal complex. So let's see how this goes here. Okay, so we're going to have an enhancer and the gene. Okay, here's our basal complex. Now there's an promoter. Let me just get a little space here. Okay, and then fairly close to promoter is an enhancer. It's called the Kruppel response element, if anybody's interested. What happens is the Kruppel protein binds to the enhancer. What was it called? Okay, what it does is it reaches over to the basal complex and directly binds to the basal transcription factor called TF2B. Now 2B, what it does is it stabilizes the TATA binding protein, and also it serves as a recruitment site for the polymerase and the TF2F that comes along with. So if you stabilize that, that means the TATA binding protein is stable, and that means you're going to much more likely be able to grab the polymerase and hold it there long enough for transcription to take place. So in this case, we get lots of transcription. But in cells that produce a very large amount of Kruppel, you see something different. So this could be in low concentration. On the other hand, when you have a lot of the Kruppel protein around, you have a totally different story. What's going to happen here? Here, we'll put our basal complex here. It turns out that that enhancer can not only just bind one copy of Kruppel, it can actually bind two copies of Kruppel. They can both fit there. But when the two copies stick together, it alters the protein structure, the complex's structure in such a way, and what happens is it actually pushes off a member of the basal complex. It displaces the TF2E, which is involved as a kinase, and that's going to prevent the polymerase from being activated and moving on down things. So in this case, the gene is not transcribed. We have strong repression. So what this point illustrates, we said many times before, concentration counts. Here's the same protein binding to the same enhancer. In low amounts, it's an activator. In high amounts, it's a repressor. So you can, just by some of these few examples, you can get a rather interesting idea of the complexity of transcriptional regulation. We've got a whole bunch of transcription factors that compete for the same enhancers. We have concentration effects. We have one protein binds to another, it activates. One protein binds to, the same protein binds to something else, it can repress. An individual transcription factor can be an activator or repressor, or a strong activator or weak activator, depending on any kind of regulation. Phosphorylation, dephosphorylation, whatever the case is. And even with the same transcription factor. A little bit goes a long way. Too much is not a good thing if you want that gene transcribed. So we have all this kind of complexity. That's one reason why we really don't have a full roadmap of how to express genes in eukaryotic cells. You not only have to look at the proteins that are going to act as transcription factors, you have to look at concentrations of them, of each one of them. You have to look at interactions of one to another. Even things on separate enhancers can interact with each other and make radical differences in transcription. So when you throw all this stuff together, you see an incredible complexity and many points to regulate transcription of a given gene. So that's the lesson for this thing. Now one last example again. Some transcription factors you can kind of call them master control genes, because what they can do is activate transcription of... What they can do is activate transcription of numerous genes, including numerous other transcription factors. And those in turn can activate more transcription factors and more other genes, and pretty soon you get a cascade of things. So you get something like a chain reaction. We have one transcription factor here, and it activates transcription of numerous proteins. I'll put boxes for some, and then circles for new transcription factors. And then these can activate transcription of numerous other proteins. And we have still more. And still more. And this whole process goes. So ultimately, a single transcription factor can lead to a chain reaction that can regulate transcription of scores, even hundreds of separate genes. And if you have the right kind of chain reaction, that can actually turn one cell type into a highly specialized cell type. It can generate a new organ, a new type of tissue that wasn't there before. So we see some of examples of that. And one of the best examples of that is the old Eyeless gene. The Eyeless gene was first discovered in fruit flies because there were mutations. Now, do they still do the fruit fly experiments in genetics where you actually raise fruit?
It's with a computer.
It's all online now.
They're going to do it this semester.
Oh, okay, I guess it depends on who teaches. Well, you know, there's plus and minus with the online thing. The plus, of course, is that you don't have fruit flies buzzing all over the department. They don't invariably get out. The minus, you don't actually get to see this stuff. And it sounds like you would rather deal with it.
It was boring. It was
Okay.
very boring.
Well, when you're actually raising your own fruit flies and these mutants and doing your own process and seeing the results, it's a little bit different than, you know, seeing stuff on the screen. Okay, that's my personal opinion. The online stuff is a poor substitute for the real thing, but, you know, you have other people, including some of my colleagues, and a lot of administrators who love that kind of stuff. It's cool, it's new technology, stuff like that. Okay, well, all right, I don't think it's a substitute. And likewise, in comparison, be watching the videos that I'm making versus actually being in the class. I mean, those might help, and some people who have used them, I don't think it's a perfect substitute, especially with crappy resolution. But hey, I get 2 gigabytes a day and stuff. Every class, 2 gigabytes. I'm going to have 100 gigabytes between cell and development of videos posted up and stored in one, stored here and stored in my home computer as a backup and all that kind of stuff. That's a serious amount of data. But at any rate, okay, so we have these master control genes. The Eyeless gene is really cool because it was first discovered in fruit flies, a mutation where the fruit flies were perfectly normal, except they had no eyes at all. They had the standard insect exoskeleton cuticle over the eyes, over where the eyes once were. So, not surprising, geneticists, hey, my God, one seems to be traced by classical Mendelian genetics, all the stuff that you hate when you take genetics, right? Okay. It actually is useful. You may hate it, but it's actually quite useful. And classical Mendelian genetics, it was very clear that it was passed on as a single trait, a single gene. You had that if you were a heterozygote or a homozygote dominant, you got eyes. If you were homozygous recessive, you did not get eyes. Ooh, okay. So one gene is making eyes. Wow. Okay, so not surprising, molecular biologists wanted to find the Eyeless gene. They did. And it was clear it coded for a transcription factor. And then the next question, this is where you use the magic of hybridization, southern blots, DNA hybridization. Remember that from our methods thing? You get a probe to a particular gene of interest, get DNA from, say, another cell, another organism, separate the strands, add in the label probe, let them seal back up again, and see if you have something, a sequence that matches the probe. Well, they did that, and sure enough, vertebrates have it too. We've got it. For us, it's called PAC6. One of a large group of transcription factors. Okay, and of course, if that's gone, there's serious ion malformations in vertebrates as well. So this is something. This is telling us something. This is telling us this one gene activates a cascade that leads to the transcription of scores, if not hundreds of genes. Now think it, and not just in fruit flies, but in vertebrates as well. Now we've got eyes, and arthropods have eyes, but arthropod eyes are very different from ours. Just a little quick thing. If you look at an arthropod eye, it's what we call a compound eye. It has, in effect, it's a whole bunch of miniature little eyes that are joined together. It's kind of like a biological CCD chip. So you get a pixelated image if I happen to be a giant fly or something like that. Remember the old science fiction thing? The fly. Okay. Well, this guy is going to have a pixelated view. It's like looking at a JPEG image, a low resolution JPEG image. As a matter of fact, the compound eyes, they're getting, for insects with extremely good compound eyes or other arthropods, you're talking about kind of like the 640 by 480 television quality resolution. That's about what you get from it. Ours is much higher, at least in very localized area. Okay, but the arthropod compound eye, what you have, the individual eye elements, you have kind of this clear tube that focuses the light, and then you have a photoreceptor cell at the bottom, and it makes a connection with the neuron. Okay, and then you have another one, and so on. And then the insect brain basically is going to process this and make a whole image. It's going to look like a low-resolution digital camera image. Okay, now when you look at vertebrates, and also cephalopods, octopus and squid have a vertebrate-style eye. Instead of these individual elements, we've dispensed with that, so we have a single lens for the whole shebang. And then we have the retina, which is a whole bunch of photoreceptor cells. And they're all connected to neurons, and then goes in, and here's our optic nerve. So it's a very different kind of arrangement. In effect, we've kind of merged, we've gotten rid of the individual elements of the eye, merged them together, and all these photoreceptor cells form a single sheet that we call the retina. Now, their eyes, they work quite well. In fact, in many cases, the vertebrate eyes are higher resolution for the most part than the arthropod compound eyes. But anyway, they work quite well. But their structure is quite a bit different. Yet the eyeless/PAX6 gene is going to trigger off events that lead to the formation of both types of eyes. Now, if you do a deletion of the eyeless gene in fruit flies, not surprisingly, no eyes form. Everything else is normal, but eyes simply do not form. Several years ago, somebody did a pretty intriguing experiment. They, with a little bit of molecular biology technique, they put an enhancer in front of the eyeless gene in cells that were of the fruit fly embryo that were eventually going to become the leg. And then, by adding a certain substance, they could activate the transcription of that particular, of the eyeless gene right over there. So what they did is they activated the eyeless gene in what was going to become the leg of the fruit fly. And lo and behold, those fruit flies ended up forming compound eyes on their knees. Morphologically, those eyes look like the normal compound eyes that you found where they're supposed to be. There were even signs of innervation. Now, the people that wrote the paper, they said, we cannot tell whether they're actually able to see out of these. We see innervation, but this is as far as we're going to carry it. Otherwise, the reviewers would beat us up real bad for going above and beyond and too much speculation. But that's a very important thing. A very complex structure that would otherwise turn into cuticle ends up as this complex structure. It's all done by a single gene. A single powerful transcription factor. So that kind of illustrates that point. Okay, well,